Good morning, grade 8. We are now in the continuation of our topic for cell division and genetics. We are now down to meiosis. We're done with mitosis last meeting. And again, mitosis is cell division that is actually for diploid number of chromosomes. And as we say that this is in a somatic cell. Somatic cells means that it has a complete set of chromosomes. Now let's move now to meiotic cell division or meiosis. Wait, let me move my screen over. Okay. Right, there you go. Meiosis. Okay. Let's move now to meiosis. What's with meiosis? Okay. If mitosis ensures that each donor cell will receive the same number of chromosomes because they are considered as diploid as the parent cell, another type of cell division ensures that the number of chromosomes is maintained among the same species. Otherwise, it will be considered what? It will be considered a defective cell. Okay. By reducing chromosome number by half, that's haploid. Haploid means half the number of sets of chromosomes in an organism's sex cells or gametes. Sex cells or gametes, we're talking about sperms and eggs. Are you still with me? I can only see 2 students here. Okay. This type of cell division is called meiosis or meiotic cell division. While mitosis and meiosis share similarities, they are different in 2 aspects. Number 1, recombination of genetic material occur, which result to variations. This is the reason why we have a variety of species, and 4 haploid cells are produced at the end of meiosis. If mitosis creates or shall we say produce 2 daughter cells at the end of each cycle, or 2 daughter cells at the end of mitotic cycle, in meiotic cycle or meiotic cell division, we actually end up with or produce 4 daughter cells with haploid or haploid number of chromosomes or half the number of chromosomes. Okay, this is to maintain again the species. Again, there are some questions in your question in your shall we say module that is asking about what is the purpose of having meiosis. This is to ensure the number of chromosomes in each species. Okay, meiosis can be called gametogenesis because it is actually the process of the production of gametes. That's gametogenesis. Okay, genesis means what? Formation. Okay, genesis means formation. Gametogenesis means this is the process of forming or creating gametes because it is responsible for the formation of gametes. As simple as that. The egg and the sperm. That's the gametes. Okay. The specific term to describe the production of sperm is called spermatogenesis because it is the production of sperm from spermatogonium or spermatogonia. That is actually the source of or the parent cell of sperms. And the production of ova or egg is called oogenesis. Okay, from oogonium or oogonia. This is the parent again of the egg, the source of the egg. Okay, meiosis results in the formation of cells that are haploid. Okay, again, the main reason why meiosis created its haploid in order to maintain, the number of chromosomes in a particular species. Okay, remember that reproduction or sexual reproduction will actually result to a zygote. Okay, this is the union of sperm and egg. So in order to maintain the number of chromosomes, okay, in that particular species, it should actually contain half in the sperm and half on the egg. And once they actually have the union or the zygote, the formation of zygote, it will become now as 1, but with a combination of sperms and the egg. Okay, that's the reason behind. Unlike mitosis, meiosis involves 2 divisions. Take note of the divisions. 2 divisions which we shall call meiosis 1 and meiosis 2. By the way, talking about your project, sure. What about a project? Okay. Remember that this project is actually your year-end project, the school year project. Okay. The school year project in the sense that it actually what? It's actually your output for the school year. Okay. Create a resource, a learning resource material out of our topic from fourth quarter. This is our topic for fourth quarter, module 1. Okay. Weeks 1 to 4, illustrating the science concepts that we had discussed. This science concept. Okay. This includes, not limited to, we have two categories for your output. The first is digital output, which includes, but not limited to, blogging, video documentaries, which is narrated. And the second category for your learning resource material is physical output, which has the biggest, again, the biggest points or the biggest grade among the outputs. So what's with the physical output? This includes, but not limited to, 3D of science concepts. It could be anything that is actually related to the topic. It should be related to the topic. 3D concepts or 3D of the science concepts, a diorama, okay, in miniatures of science concepts, or the actual science concept if possible. So you just choose between digital outputs or physical output. But I am encouraging everybody to please do the physical output. Why? Since number 1, it has the biggest grade. And number 2, it will be published or it will be exhibited during the time that you are allowed to do so. I will actually put that one also in the laboratory, okay, in the science lab. Make it as attractive as possible with your own creativity and any materials of your choice, okay? Any materials of your choice. It doesn't necessarily mean that you will buy the materials. You can actually maximize the materials that you have at home, okay? Take note that this is your output for this school year, okay? And again, it will be exhibited as soon as we are allowed to do so. We'll be in school every Friday to check on your outputs. And the deadline for this, heads up, this should be submitted on or before May 13, 2020 or 2022. Okay? On or before May 13, 2022. Today is April 6, 2022. Meaning to say that you have ample time to prepare. You have ample time to choose between the topics inside that particular topic, inside this module. You can choose any concepts. And this is more than a month. This is more than a month. This is 1 month and 1 week in preparation for your project. So most probably you can do, or you can search a lot, if in case. What is a learning resource material? Talking about learning resource material, these are materials that actually expound or explain the concept. Okay, I will give you one example. Are you still with me? Are you still with me? Can you hear me? Okay. This was created by my previous students. Okay. This is the learning resource that I have here. Okay. This is under circulatory system. Can you see my shared screen now? Can you see my shared screen? All right. So this is a 3D heart. As you can see, okay, in the legend, you can actually see there, the blue one indicates carbaminohemoglobin. So the blue one indicates that it actually carries, or this blood carries, carbon dioxide or carbaminohemoglobin. And the red one indicates oxyhemoglobin. So this actually carries blood with oxygen. So as you can see, it explains the concept and also the parts of the heart. So if you're gonna open this heart, you can actually see the blood flow, okay? And the direction where the blood actually flows with the labels. The labels also indicates what? The parts of the inner heart, okay? Which explains the concept. So this is one example of a learning resource material. This is the one, or this is actually under the 3D, the three-dimensional learning resource material. So it actually explains the concept of circulatory system, okay? Talking about heart. This is the heart, the human heart, right? Hopefully we can do also more or less more than that one, of course. And our topic, this should be based on our topic, okay? Cell division and genetics. Do you have any question for with regards to your project? Question so far? Greg, Tisha, and Kalina? Question? Are you still with me? Can you hear me? Yes, yes, Kisha. Correct. You should pick a portion of that lesson or the lesson itself because you cannot actually create more than that one. Like for example, if you're gonna choose mitosis meiosis, then you can do so. And you can create a resource material, okay, a learning resource material out of it, which will explain again the concept of cell division and genetics, or it explains the concept of mitosis meiosis. Okay. Any other question? Okay, thank you for asking question, Kisha. I really appreciated it. Any other question? If we pick a video discussion, it will be in any style. Yes, it could be in any style as long as it is narrated. It could be documentary, it could be in a blog form, as long as it is narrated by you. And of course, with good outputs, with good videos that explains the concept. It could be, yes, correct. Any other question? But again, the biggest grade belongs to the physical output, of course, because we can actually preserve that one and put it in the laboratory, the science lab, and have it exhibited after. If DepEd will give us a chance, why not? Or be putting it in an exhibit. Any other question? All right. So let's move now back to the lesson, the meiosis 1 and meiosis 2. These are the two main divisions of meiosis. So let's tackle meiosis 1. Meiosis 1 is reduction division. What's with the reduction division? Okay, let's take a look at this one. If mitosis has prophase, metaphase, anaphase, and telophase, meiosis also has prophase, metaphase, anaphase, telophase. But it depends upon the what? It depends upon the divisions that they actually belong. Like for example, if it is in meiosis 1, prophase 1, it is actually prophase 1 because it belongs to meiosis 1. Okay, prophase 1 is pretty much extended in meiosis. Okay, at this stage, chromatin condenses to become shorter and thicker chromosomes. Homologous chromosomes, those that carry the same type of information, pair with each other, lie side by side to form tetrads. Tetrads means tetra. Tetra means 3. Okay. It is during the formation of crossing over. This is crossing over, by the way. So crossing over of genetic characteristic. This is the reason why we are actually diverse. We are actually what? More. We have more variants. Okay. We are in a variety in a particular species. This is the reason why. Because of crossing over. Crossing over between homologous chromosomes allows shuffling or exchange of genetic materials. Okay, and produces variation of offsprings or in the offspring. Similar to mitosis, the nuclear envelope begins to break down because it is in prophase. Remember that prophase is breaking down. Okay, breaking down while telophase is actually building up. Okay? The nucleus disappear, the centrioles move in the opposite poles. Okay? In metaphase 1, at metaphase 1, this is the second phase of meiosis 1, homologous chromosomes move to the equatorial plate. If it is in metaphase, it should stay at the equator line or equatorial plate, okay, with the centromeres attached to spindle fibers and oriented on the opposite poles. This is now the time that the centromeres or centrioles are actually pulling, okay, the chromosomes apart or away from each other, but they are actually in line in the center equatorial line. So if you're going to have an illustration, this is prophase, okay, metaphase at in the equatorial line, metaphase, anaphase, and telophase. This is also cytokinesis. Okay. During anaphase 1, this is the third phase of meiosis 1, the homologous chromosome separate and moves towards the opposite pole. Okay. Unlike mitosis, sister chromatids remain together in anaphase 1, and the cell membrane begins to cleave. There is a cleavage in anaphase 1. Okay. In telophase 1, okay, this is the last phase of meiosis 1. The nuclear envelope begins to form. And this is now the what? The building up stage. Form around the chromosomes, sister chromatids, and eventually ends in the cytokinesis, forming two daughter cells with half number of chromosomes as the parent cell. Okay? It actually creates now two daughter cells with half number of chromosomes as the parent cell. Okay? So that's the meiosis 1. If you're going to look at it under the microscope, okay, this is meiosis 1. Okay? This is really under microscope. Okay? You can actually see pairs here with homologous chromosomes, condenses. Okay? And crossing over happen with the same traits. They actually share what? Share the uniqueness of these traits. Okay? In the metaphase 1, as you can see, they are actually aligned in the equatorial plate. Okay? And now, the centrioles are actually at the opposite poles with the spindle fibers over here, okay, pulling these chromosomes on a separate or in different plates or in different areas. They're pulling away from each other, as you can observe. On the anaphase 1, as you can see, there is a separation. Okay, there is a separation of different sets of chromosomes. And in the telophase 1, as you can see, cleavage is formed. Actually, in anaphase, there is a cleavage formed, and it also builds up. Okay, this because telophase is building up, builds up the covering or the membrane, the cellular membrane of that particular cell. We have now two daughter cells. That's in the meiosis 1. How about meiosis 2? The equational division. What's with the equational division? Meiosis 2 begins with prophase 2. Okay, because this is now the second division of meiosis. Okay, equational division. Meiosis 2 begins with prophase 2, whereby the nuclear membrane breaks up. Again, breaking up is in the prophase, building up is in the telophase. Breaks up the spindle fiber, breaks up and the spindle fiber forms. The sister chromatids no longer replicate, but begin to migrate in the equatorial plate. Okay. In metaphase 2, sister chromatids are actually at the center of the equator or equatorial line. Okay. In anaphase 2, these sister chromatids split more towards the opposite poles. And in the end of telophase 2, a new nuclear envelope form around the chromosomes. The nucleolus reappears and the cytoplasm divides. That and end with 4 daughter cells with each half the number of chromosomes as the parent cell. Okay? As you can see, this is meiosis 2. In prophase 2, as you can see, the cellular membrane breaks up, okay? And the centrioles move in opposite direction in preparation of pulling, okay? Spindle fibers are actually attached to the centromeres of these chromosomes in metaphase 2. Still, they are aligned on an equatorial line. And the pulling, okay, the pulling of these chromosomes is in the anaphase. You can actually see some cleavage there. And the telophase happens when they actually separate. Okay, the building up, they build again another what? Cellular membrane. And we end up with 4 daughter cells. So the result of mitosis is 2 daughter cells, while the result of meiosis is 4 daughter cells. Okay, do you have any questions so far? I think we're running out of time. Hopefully you learned something out of a simple discussion. By the way, again, you can use these concepts in your learning materials. Okay, you can use this concept in your project, okay, the learning resource material, and hopefully you can have brilliant outputs for this school year. Take note again that this is an output for this school year. I didn't ask for any project in the previous quarters, since I am preparing you for this, but I am giving you a month, more than 1 month, to do this project, right? Do you have any questions so far? Questions so far? Greg, Keisha, Kalina? Can you still hear me? All right, I think that will go for today, class. Thank you for participation. Hopefully, we can do greater than those with my previous students, an output for school year. I think that will be all. Have a nice day, grade 8. Take care. Bye-bye. Enjoy the rest of the day.